We began our unit talking about viruses and vaccines, and now we want to move on to the second part of our unit by talking about bacteria and antibiotics. Bacteria are prokaryotic cells. Prokaryotic cells are a very simple type of cell that do not include a nucleus and do not include any other organelles. Animals and humans are made out of eukaryotic cells, which are much more complex, featuring a nucleus and also various organelles. In addition to being prokaryotic, bacteria cells are very small. They're approximately 1/10 of the size of other cells. The picture at the right shows us that bacterium are very small, approximately 1 micron, but reminds us that compared to viruses such as the pox virus and influenza virus, bacteria are actually somewhat large. Another feature of bacteria is that they have no internal compartmentalization, so no nucleus or anything like that. If we look at this diagram, you can see they have just this loop of DNA, they have some ribosomes, and then they have their cell wall and flagellum and pili. Prokaryotic cells are also unique in that bacteria do not work together. They are unicellular. That means that they do not depend on other bacteria cells to exist. In our bodies, we have complex networks of eukaryotic cells that coexist and allow us to function, but bacteria can function and survive independently of each other. Bacteria also have chromosomes, just like eukaryotic cells do, but they have a single circular loop of DNA that is not contained in a nucleus. Not all bacteria move on their own, but some of them do, and they use something called a flagellum to propel them through a medium. They also have pili, which allow them to attach themselves to surfaces, which can allow them to stay put and thrive more successfully in an environment. Bacteria reproduce using something called binary fission. This is when one cell pinches into two cells and divides. When this happens, the chromosome is duplicated, the cell grows, and the duplicated chromosomes separate, and then the cell divides itself in half, similar to the end of mitosis. And then we have two new cells with a complete set of chromosome. Many bacteria can complete metabolic activities, and that means they can get their food and nutrients from a variety of sources. So they can be anaerobic or aerobic, meaning that they can either use oxygen in order to metabolize nutrients, or some of them are capable of metabolizing nutrients in low oxygen environments. There are three basic shapes that bacteria take. The first is circular. If a bacteria is circular in shape, we would say that it is called coccus. And so we often use this as the ending to designate the shape of a bacteria in its name. This is a picture on the right of some circular bacteria. So this would be a coccus bacteria. Bacteria can also be rod-shaped, and we call this bacillus. Bacillus has this slightly elongated shape that almost looks like a capsule or something like that. Corkscrew is the final shape of bacteria, and we refer to that as spirillum. The spirillum is a little bit bigger than the other shapes of bacteria, mostly because it's very long and stretched out in its spiral shape. In addition to coming in multiple shapes, bacteria can also occur in different groupings, and that's another way we can classify them. So the groupings include diplo, which is 2, a cluster, sort of like a cluster of grapes, which we refer to as staphylo, and a chain, which we call strepto. And the combination of the shape and the groupings allows us to name a bacteria. So let's do a couple examples. So if we have these round bacteria and they're all grouped together, we identify the shape as being coccus and we identify the grouping as being staphylo. And so it would be called Staphylococcus because it is round in shape and clustered together. The next grouping that we could look at are these little rod-shaped ones, which we call a bacillus shape. And then we could also see that we have a chain of them and that there's multiples. And so we would call that a bacillus, strepto, and we would put those together to get streptobacillus. This last guy includes the Spirillum, screw-shaped bacteria. And again, we have multiples of these that are kind of in a chain, and so we would call it Spirillum. The grouping would be strepto, and so we would call it Streptospirillum. And so when those terms are used, we're referring very specifically to how the bacteria actually look when we observe them under the microscope. Bacteria actually can have two types of cell walls, which is pretty nifty. And we can use that to gain information about what kind of bacteria it is. And sometimes we can use that information to figure out how we can destroy the bacteria to return someone to health. So cell walls are determined by something we call a Gram stain. And in a Gram stain, we use dye to try to color the bacteria cells to identify which is which. So if something is Gram positive, it will stain violet, and we know it has a certain type of cell wall. If it's Gram negative, then the stain will turn the bacteria pink. And you can see in this picture that we have the violet, these dark ones, that are Gram positive, and then these pink ones that are Gram negative. And so this is very helpful when we're trying to identify antibiotics to use to fight a bacteria, because some antibiotics work on Gram positive while others work well on Gram negative. So this is a very powerful test for physicians and scientists who are trying to improve health. So we could take a look at a couple pictures and try and decide which is Gram positive and which is Gram negative. So we would look at the color, and we could see that the ones on the left are pink and the ones on the right are purple and dark, and we could correctly identify the left-hand side as being Gram negative and the right-hand side as being Gram positive. Bacteria, like all cells and living things, need to metabolize food sources into energy so that they can survive. And there's three ways that bacteria can get their energy. And a couple of these are actually really cool. So bacteria can be photosynthetic like plants. They get their energy from the sun and use it to build molecules like sugar that they will later use for energy. A heterotrophic bacteria gets its energy by eating and consuming other organisms, which, if you think about it, is what humans do. We eat plants and sometimes other animals to get our energy. There's also chemoautotrophs, and these types of bacteria get their energy from inorganic minerals, so things like soil or water, which is really cool. That's not a source that we as humans are able to tap into. We can't get our energy from water. That would be cool because there's lots of water on the planet, but that's not really how it works. Additionally, with bacteria, we want to talk about something called an endospore and a pyle. So endospores are a type of bacteria in hibernation. So if the conditions are too harsh for the bacteria to live, they form these hardy little spores and they wait until the conditions are more favorable to try to live and reproduce. Pylae are little hairs on bacteria that allow them to stick to surfaces, and it forms bridges between bacteria to exchange genetic information. And this is how bacteria become resistant to antibiotics, because if one bacteria is able to survive the antibiotics because of some genetic information that it has, it can actually use its pylae to transfer that to other bacteria so that they can also survive the antibiotics. Here's a picture of all of the pylae connecting different bacteria. So bacteria can actually be helpful or they can be harmful. So we're going to talk about a couple examples of each of those things. Harmful bacteria can cause disease in two different ways. The first is to metabolize their host. So they actually are just eating their host and using that for energy. Tuberculosis is an example of that. Tuberculosis is a dangerous lung infection where the bacteria is inhaled and settles in the lungs and grows on human tissue. If untreated, the patient generally languishes and dies within 18 months. And this is a picture of a tuberculosis patient. You can see that they're very emaciated because all of their nutrients is going to the bacteria instead of into their body. And it's a very horrible disease for that reason. Another example of bacteria that's harmful is the bacteria in our mouths that causes tooth cavities because it destroys the minerals making up our teeth. Lyme disease is a harmful bacteria that is the result of being bitten by an infected tick, and it causes a rash, pain, and swelling in the joints. So on the left, we have that little tick, which is disgusting. And over here, we have the typical bullseye rash pattern that you see with Lyme disease. And the horrible thing about Lyme disease is that we don't have a cure for it. All we can do is try to treat the symptoms and help the patient as they experience pain and swelling in their joints. The second way bacteria can cause disease is through secreting toxins. So an example of this is diphtheria. Diphtheria used to be a very serious issue, and it grows in the throat but produces toxins that attack the heart, nerves, and liver. You can see in this patient there's diphtheria growing at the back of their throat, but it would ultimately kill people by undermining their heart, nerves, and liver with the toxins that it creates. Food poisoning is perhaps a bacteria that we've all experienced, and it causes an upset stomach and gives us different problems when we've eaten food that has this type of bacteria in it. Botulism is another bacteria that causes disease in food, and it produces these endospores and it grows in canned foods, and when they're improperly heated, prior to sealing, the endospores have a chance to thrive, and this can actually cause paralysis and was a very real problem during the Industrial Revolution. Fortunately, we've had a lot of advances in technology and medical knowledge that have allowed us to eliminate the problem of botulism. The best ways to fight bacteria include washing your hands with soap to actually physically remove bacteria from your hands. You can use chemicals to kill them or antibiotics. Alexander Fleming found that bacteria didn't grow around a particular fungus called penicillin, and this was the first antibiotic that we ever had and was very, very effective against pneumonia, which was another dangerous infection of the lungs. Antibiotics destroy cellular components of bacteria. And so you can see in this picture that here where the penicillin is growing, this bacteria is like, oh no, we're not going near that, and stays far away. Antibiotics cannot kill viruses, however, because viruses do not have any cellular processes.